Good evening, everyone. Welcome to Botany Insider's new free session for the PYQ pointers solution. Let's wait for everyone to join and then we'll start off with our class, right? Till then, those of you who are there in the class, please confirm in the chat sections whether the audio and the video is clear to you or not. We'll be discussing about few more questions of June 2023 morning shift paper. If you remember, in the last class, we discussed few questions already. So make sure that you are following up the previous classes as well, because we will be completing a number of questions in the entire series. Okay. Good evening, everyone. Good evening, everyone. Good evening. Make sure you come to these classes on daily basis. We'll be discussing questions related to the same. Okay, so let's begin with our class. Others would join in the time being. First of all, make sure that if you're preparing for the December examination and if you're lacking any structured preparation and if you feel like that you need to have a structure of preparation with yourself, please make sure that Botany Insider have its own application and you can enroll in the two courses running right now. One is a full course and one is a crash course. Okay. In both of these courses, you'll be getting hard copy notes. In the full course, you'll get the detailed ones, and in the crash course, you'll be getting the revision notes. Okay. So if you wish to enroll in the same, you can download our application and get yourself enrolled. We also have option for buying the notes alone. So you can WhatsApp us on this particular number so that you can get your notes sample and then you can decide for ordering them. Right, let's begin. In addition, we are about to start the enrollments for the PYQ course for the December 2023, in which we are going to discuss the previous year questions in the most detailed manner possible. So make sure that if you want to have just a course for the previous years, because if you look into the previous year questions, like if you look into the June examination, which was there on 6th of June this year, you must have seen that a number of concepts got repeated as it is, right? So doing the previous year questions is of utmost importance and it becomes a bit tedious task to do it yourself. So you can just enroll in this particular course. We are about to start the enrollments soon. Okay, so this was about it. Let's begin with our class now. This is the very first question for you all, everyone. The question is, given below are few of the characteristics for your shade plants and for your sun plants. Okay. Question is asking you that you are given few of the options and you need to find out for a plant which gets acclimatized for your shady region or which can easily grow up in the shade region, which all characteristics will this particular plant showcase. Very easy, very important. Give it a try to the question, everyone. Okay. Give it a try to this particular question. Just a second. Yeah. Give it a try to this question, everyone. Very easy question. You're given few of the statements for the sun and the shade acclimated leaves. You're given few of the statements and you need to find out which of the following is correct in terms of your shade acclimated plants or the shade acclimated leaves. Okay. That means those leaves which can easily perform the various functionalities while staying in the shady region as well, where they're not getting very optimum level of sunlight. Give it a try to the question, everyone. And this was June 2023 morning shift. And exact same question got asked in your previous year as well. So you can understand doing previous years is a very, very important part, right? It's a very important part for your preparation. First option is high dry mass per unit area. Second option is high number of chloroplasts. Third option, low chlorophyll A to chlorophyll B ratio. What is the meaning? Chlorophyll A to chlorophyll B ratio is lower Like the overall ratio is lower. When the overall ratio is lower, that means what? That means your chlorophyll B is more, chlorophyll A is less, right? That's why we are getting the overall result as lesser value. Then if lower dark respiration per area, then you have higher light intensity complexes per area. Give it a try. If you go in a logical manner also, you'll get it very easily. Give it a try, everyone. It's a logical question also. If you have done plant physiology properly, you'll get it. Even if you get it incorrect, that's absolutely fine. Okay, you have to make sure that you are trying it out. Because in your preparation phase, in your practicing phase, you should not fear from wrong answers. Give it a try, everyone. Very simple question. Very good, everyone. Give it a try. Mark the answer. 16 of you are there in the class right now. One more minute, then we'll solve it. Done, everyone? Okay, let's see. So the question is that you're given few of the characteristic features where you're given the information related to your shade acclimatized leaves. Okay, and you need to find out that which of the following are the correct characteristics. Answer for this particular question is option number B. Okay, that is your C, D and E is the correct answer for this particular question. Remember, they are asking you about shade acclimated, that is they are not adapted. There's a very big difference between acclimatized and adapted. Adaptation is on the gene level. Acclimatized leaves are the leaves, or acclimatization means that for a very short period of time, that particular plant or that particular organism is behaving in a different set with a different set of characteristics. Okay, so low chlorophyll A to chlorophyll B ratio, lower dark respiration per area, and then you have higher light harvesting complexes per area. The answer for this particular question will be option number B, according to the CSIR's official answer key as well. Okay, so chlorophyll B to chlorophyll A ratio higher. See, chlorophyll A to like chlorophyll B to chlorophyll A ratio higher. That means what? That means this is less, this is high. Okay, you have to keep a note of this point as well, and you have to keep a note of the like the what is asked in your question. They're saying chlorophyll A to chlorophyll B ratio lower. Okay, over here it is written chlorophyll B to chlorophyll A ratio higher. These two things are same. Reason being, chlorophyll A lower, chlorophyll B higher. Okay, and the overall result will be lower. Okay, that's why answer for this is option number B. I hope the concept it's a very elaborated concept that we study in plant physiology, right? So make sure that you're getting this concept very much clear in mind, right? Next question. Now the question is related to that which of the following or what is the meaning of AmphiZone? This question is from cell biology and specifically from your plasma membrane concept. Okay, give it a try to the question, everyone. Very easy question. It is asking you which of the following describes your AmphiZone. Okay, first option is that it is a intermediate hybrid organelle that is produced through the fusion of endosomes with autophagosomes within the cells. Then it is a double membrane sesquipedal vesicle, that is a vesicle that is a hallmark for the intracellular catabolic processes known as your macroautophagy. Then it is a compartment that is fanned when autophagosomes fuses with a lysosome. Last, it is a vacuole that arises when the membranes of the ER sesquiter the parts of the cytoplasm. Give it a try to the question, everyone. Very simple question. Give it a try. Then we'll solve it out together. What do you mean by an AmphiZone? If you know the concept of the like what plasma membrane is, what is the entire concept behind the same, you'll be able to mark it. Very good, very good. Even if you get incorrect, absolutely fine. No worry in that. But mark the answer. Try it at least. Very good, everyone. Done, everyone. Very simple question. AmphiZome. Let's look into the answer. Yes, AmphiZome is what? It is an intermediate or a hybrid organelle that is produced via a fusion of an endosome and a autophagosome, right? Remember about this particular thing. Very simple question. And this was a 2-marker. So you can directly get the answer for this. One more question. Now, this one is from the concept of transposons. Okay, this is one of the concept in your fundamental processes. That is unit number 3. Give it a try to the question. You need to find out which of the following statement about lines that are present in the human genome is true. You need to find out the correct statement. 4 options in front of you. Give it a try to the question, everyone. Now, remember that this concept of line has been repeatedly asked from 2020 to 20 till 2023. In every single year, this question has been asked, this concept has been asked. So, make sure that if you are preparing by yourself, you are not skipping this concept. Okay, very easy and a very important concept. Get the answer for this, everyone. Done, everyone. Mark the answer for the question. This is related to your transposons. First concept is, or the first statement is, lines preferentially localize at the AT-rich DNA. Then lines cannot transpose independently. Then parasitizing on the line element, transposition machinery, line can attain a very high copy number. And the fourth one is the ALU family is the most prominent line family in terms of the copy number. Give it a try. If you remember about the facts for the line and the complete concept, you can easily mark the answer. Give it a try, everyone. 17 of you are there in the class right now. Very good, very good. You need to find out the correct statement. Okay, remember, while marking down the answer in your examination as well, you are very much like focusing upon the word correct, incorrect, true, false. Okay, because with just reading the question properly, you increase your chances of marking the answer correctly. Right, let's look into the answer. The answer for this particular question is option number 1. Line preferentially localized at the AT rich DNA. See, this statement is incorrect for sure. That is, line cannot transpose independently. Then parasitizing on the sign, they can attain a high copy number, incorrect. Then ALU is the most prominent in terms of copy number. No, ALU is not the most prominent one. You need to remember the various facts. Answer is option number 1. See, if you look into this particular question and if you go into like whenever you are doing your revision and everything, make sure like after the class also try checking your facts for this at least once, because answer for this is option number 1. Remember about this. Next question. Now this was a, we can say, general knowledge question that was asked in the examination. Okay, question is, which of the following species of birds is known to migrate across the Himalayas? Okay. Which of the species of birds is known to migrate across the Himalayas? This is something that you don't study while you are preparing. This is something we don't like, obviously, in your notebook or in your syllabus, it's not written down to learn the migratory birds and the at which place they are going, right? So these questions are something based on the general knowledge. So if you know the answer, very good. And if you get such question, if you come across such question in the examination, it's better to like fuck off with these and just let these questions be passed by, because there are higher chances for you to get negatives in these questions. So make sure that, and remember, in examination, you'll be getting every type of question. It could be a complete mix of variety which you'll be getting, right? So you just cannot stick by or think that only from the syllabus you'll get the questions. No, you'll get questions from the general knowledge part as well. Try out this question if you know. But also in your PYQs, many times questions get repeated. Okay, in the previous years, if you look into the questions, you see a number of questions getting repeated. So whatever information you can take up from the PYQs, take it up and make your own pointers in your notebook. It's a very, very important exercise to make PYQ pointers. Okay, done everyone. I guess very less students have knowledge about this, because obviously it's a general knowledge. Answer for this is bar-headed goose. Okay, so it's the bird which migrate across the Himalayas. This is how it looks like. Okay, remember about this. This is bar-headed goose. So this is how the bird actually looks like. Keep a note and remember about this particular information for your upcoming examinations, right? I hope the question is clear. Very good, Komal. Thank you so much for this information. Everyone, keep a note of the information that she has shared in the chat section. Next question for you, everyone. Give it a try to this question now. Segregation of the alleles that occurs either at the anaphase 1 or the anaphase 2 of the meiosis, which is the ideal model system that is utilized for studying the allelic segregation? Very, very simple and a very straightforward question. Remember about this particular question, and it's something that if you read the syllabus of the genetics unit, you'll get it. It's so simple. Segregation of the alleles that can occur either at anaphase 1 or anaphase 2. Question is asking you which of the system is the ideal model system that is utilized for identifying that what is the stage or at which stage do we have the allelic segregation getting done. Very good, Sona. Others also mark the answer. Basic, straightforward, simple question. This was a part B question. You can see you get easy questions as well in the examination. Mark the answer for this. Question is related to your allelic segregation. It is not related to your gene level. Like they're not talking about def bio. Drosophila is the model organism in your developmental biology. Question is related to your allelic segregation. And this was the same question that was asked in your, I guess 2015 or 16. There was the exact same question got asked, which was related to writing down the name of the model's organism. Arabidopsis, no. Arabidopsis is majorly for your ABCE model of flower development. Answer for this question is Neurospora carsa. Answer for this is Neurospora. Ordered and unordered, tetrad and octad preparation or studies are done with the help of Neurospora ecrasa. Remember, in genetics, you use Neurospora as a model organism. Arabidopsis is for ABCE model. Uh, Drosophila is a model organism for your developmental biology. Every particular individual, like all of the ones written down over here, all of them have a specific role in different biologies. Okay, remember about this. Neurospora is for your genetics. Keep a note. Next question, give it a try to this. The question is related to your AVR proteins. Okay, question is related to your AVR proteins. Now, over here, you have to be very uh, cautious about the T4SS and T3SS. The type 4 secretory system is found in your, when we study about agrobacterium mediated gene transformation. So over here, they are talking about type 3 secretory system. Okay, don't get confused. Give it a try to the question, everyone. First one is AVR protein predominantly have SECA secretion signal. Option number B is AVR proteins. They are secreted through type 3 secretion system. Then you have hypersensitivity response and pathogenicity cluster proteins. They are involved in the secretion of AVR proteins. And fourth is some of the components of the T3SS type 3 secretory pathway. They are conserved between animal and pathogenic bacteria. Give it a try to the question, everyone. Very simple. Again, try to mark the answer for this, everyone. Okay, let's look into the answer for this question. The answer for this question is option number 1. That is AVR proteins predominantly have SEC secretion signals. This is a incorrect statement. Except for this, all the other 3 statements are correct. Okay, now with this question, let me give you one important suggestion. See, you'll get a lot of question in the examination about which you might not have studied. Okay, there'll be a lot of questions. Like have this particular thing clear in mind and make your or keep your mind prepared for this. First thing. Second thing, make sure during your practicing time, during your preparation time, you are not skipping those, like not even a single question to be skipped. See, this question, if you don't have knowledge about this, the best way, like if you are sincere about your preparation, the best thing that you should do is you should take up this particular word, AVR proteins, write down in your notes that what AVR proteins are. Take these 3 informations, these 3 pointers and note it down. In addition to that, read about what AVR proteins are. Google it there. Like you have pool of information. Like Use chat gpt, use open sources as much as possible, and read about what avr proteins are. Utilize your free sources as much as possible. Okay, because in the examination, CSIR is not going to give you the questions in a bowl and they'll ask you to just pick up a green ball or a red ball. No, that's not the way CSIR is behaving now. It's like they have changed the pattern of the examination and they have raise the bars very very high, right? And you have to change your tactics, you have to change your way of preparation. So utilize your previous year questions in or as a source of your preparation. This is what I did during my preparation time as well. See, every student goes with the syllabus part, right? It's not like that if 10,000 students are sincerely preparing, see, almost a lakh student give the examination, but at max 10,000 to 20,000 are sincere about their preparation. Every student have syllabus of CSIR in their hand and everyone would be doing that syllabus, right? What is the difference or what is the extra edge point that you are doing? That should be utilization of the PYQ. And I'm telling you, not many students do it this way. If you do it that way, you'll be a rank holder. I'm telling you, because that's how preparation is done. Preparation is not about going on a path, just following those 13 units and you will get the examination. No, you have to be smart enough. You have to be smart, be very much ready for working hard in the best manner. Okay, so don't leave the questions. I have started off with this particular thing over here just for you to get a glimpse of it. Okay, we'll be having a complete PYQ course in which every single question will be dealt. Okay, every single question will be dealt. So if you're doing it by yourself, it's absolutely fine. You can do it that way as well. Make sure that you're doing something to stand out. Okay, make sure that you're doing something or the other to like completely changing your way of preparation so that you can have an extra edge in the examination. Okay, this is a pro tip which you can have. Like if you remember, or because we have one more educator, Khushi ma'am. She was a student initially with me and she got a good rank. She got all India rank 88 just because she had an extra edge in her preparation. And this was the strategy which she used, that is using all the questions, all each and every previous year question as a source of preparation, not just a question, but taking the question and using it as a source of information addition. Okay, so do it that way. One more question for you all. Give it a try to the question, everyone. Question is, which of the following is the best suited method to estimate the population of fishes? This question is from population ecology and like unit number 10, ecological principles, and the topic is population ecology. Very, very simple question. First is camera trap, second is line intersect, third is point count, and fourth is mark recapture. Give it a try to the question. Very, very simple question. Just recall the name of the trick, recall the name of the method, and you'll get the answer. Very simple question. Very good, very good. Excellent, everyone. Yes, the answer for this particular question is mark recapture technique, right? We have discussed like those students who are there in the full course and the crash course, you can remember and you can recall. We discussed about the mark capture recapture technique in detail. The complete formula and everything was discussed. Write down the formula for the mark capture recapture technique, everyone. Those of you who have studied this, what is the formula for this mark capture recapture technique? Yes. What is the formula for this? Finding out the final population. It was sample 1 into sample 2 divided by your marked individuals that we got in the second recapturing, right? S2 was equal to marked plus the unmarked individual, right? We had a discussion about this. I hope you're able to recall. And those of you who have prepared yourself, I hope the concept is clear, very much clear to you all. Right. So these were the questions for today. From this question, we'll be starting like from the next class, in the next class. Okay. I hope all the questions and everything is clear. So this was about this free session. If everything is like, if you feel like that these sessions are helping you out, you can download our application and get yourself enrolled in the courses that we have run that they are running right now for the December batch specifically. So we have a full course and a crash course. In addition, you can get your PY notes ordered as well individually, and this course is about to start very soon. Okay, so this is about it. All the best, good luck, take care everyone. Okay, bye everyone, take care.